finishing up on the comparison here with RNA and DNA. Again, RNA is normally double-stranded and DNA is normally single-stranded. And in terms of their sugars, DNA has deoxyribose and RNA has the sugar ribose. But otherwise, they're very similar structurally and chemically. So let's move on to Watson and Crick's discovery of the double helix. And like with most discoveries in science, it's more people involved, sometimes totally behind the scenes. So James Watson and Francis Crick determined that DNA is a double helix structure composed of two polynucleotide strands, and that's what we see here, double helix structure. And they got help from, importantly, some X-ray imagery of the DNA molecule from Rosalind Franklin, who was working at a university in the lab for King's College for Maurice Wilkins. So these two people contributed quite a bit for giving evidence to suggest a structure of the DNA molecule. So the DNA molecule, as determined by Watson and Crick with help from Wilkins and Franklin, who contributed information, was that it is a double helix structure analogous to a twisted ladder, okay, into a spiral. So the sides of the ladder represent the sugar phosphate backbone. You can see here where you see a number of red and orange molecules along with the gray. And then the rungs of the ladder, which you would step on and handle if you were going up the ladder, represent the base pairs that are connected by hydrogen bonds. You notice the little gap between them as this spins or rotates. That indicates the hydrogen bonding between the base pairs of the nucleotides. If you remember, the sugar and phosphate components of the nucleotides are bound by covalent bonds. Remember, covalent bonds are considerably stronger than the weaker hydrogen bonds. So the DNA model is like a ladder twisted into a spiral. And this is just what's often referred to as a ribbon type model of the DNA molecule. And this is a space field model of the DNA molecule. Notice that it's kind of spirally twisted around. And again, the sides represent the sugar phosphate backbones, and the rungs represent the hydrogen bonded bases of the nucleotides, right? And the backbone, that's the sugar phosphate groups, covalently bonded to each other. And the base pairing is dictated by the complementary base pairing rules, in the sense of, if you remember, A always binds with T and G always binds with C, which you can see over here on the left. So because of their pretty monumentous discovery, they received the international notoriety or acclaim. Watson and Crick, in 1953, they published their results in a relatively short paper suggesting this double helix structure, spiral double helix structure, as the actual molecular shape of DNA. And then a few years later, in 1962, Watson, Crick, and Wilkins received Nobel Prizes for their discovery and presentation of the DNA molecule as that double helix. And you probably haven't heard of Franklin's name or even Wilkins' name before, because, for one, Franklin died in 1958 due to cancer, and for whatever reason, the Nobel Prizes are not given posthumously. In other words, after death, they do not award the Nobel Prize to people. And why they do that, I have no idea. So a little video here on the double helix structure of DNA. Get the volume up. There we go.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
All right, a few different representations of the double helix structure. So in their 53 paper, Watson and Crick wrote that their structure of the DNA molecule immediately suggests a possible copying mechanism of the genetic material. And I mentioned this before. Because of the complementary base pairing, if you were to separate the two strands of DNA, so you had a single strand, because of the complementary base pairing, it's pretty straightforward to match in the correct nucleotides to reassemble the missing half, because you'll always know that a T base nucleotide binds with the A base, a G base containing nucleotide always binds with the C base, and etc., and etc., etc. So because of that complementary base pairing, that is the real key component to being able to copy the DNA molecule. So the structure of the DNA molecule gives a molecular explanation for the unique characteristics of the DNA molecule in its ability to copy, reproduce itself, and pass on that genetic information to the offspring, whether they're cells through mitosis or sexually reproduction offspring through sexual reproduction. Think of meiosis. So when a cell reproduces, it must copy its genetic material DNA before the cell divides. Again, think of when we covered mitosis and meiosis. And then the copied genetic material, the two copies of the DNA molecule, one copy goes into each daughter cell. Again, think of mitosis. Go back and take a quick look at that. So Watson and Crick's, their DNA model shows that each DNA strand, here we see a complete DNA molecule, double helix, and it's zipped apart, and the two halves represents one single strand of DNA. So the single strand of the DNA molecule serves as a model or a mold or a template to guide the synthesis, reproduction, copying of the other strand, as we see here, occurring through the action of this enzyme called DNA polymerase. So a few of the enzymes we'll need to know. One is called helicase, which unwinds a double helix, splits it apart, breaks those hydrogen bonds between the complementary matching bases of the nucleotide. So then the DNA polymerase enzyme, remember enzymes often end in ase, is able to come in and see that, oh, here's a T base. It's supposed to match complementary with an A base, and it brings in that A base containing nucleotide there. And then another one here, A, and then another one, different one here, C, and then here, A, and then here, G. And because of that, complementary base pairing, the replication part of DNA is actually fairly straightforward. Okay? Here's a figure from your textbook. The original strand can be unzipped. So we'll talk about the parental and the daughter strand once the original DNA molecule is synthesized or produced off of each single strand template and color-coded in blue. So here's the original intact DNA molecule. It unzips, unwinds through an action of an enzyme, helicase. And then another enzyme, DNA polymerase, comes in and links in the appropriate nucleotide with the complementary base. And that new strand is color-coded here in gold. The original template strands are in blue. So once the new entire DNA molecule is synthesized, 2 copies, 2 copies to go into each daughter cell. So there's the daughter DNA molecules, the double strands, double helices. Each one contains a parental strand, an old strand from the original DNA molecule that got copied, and a new strand, or a daughter strand here in gold, comprised of the individual nucleotides that were brought in to bind with the lone bases on the template strands. All right. So little